Hello again, biologists. Mr. Kinnear here with another video lesson for National 5 Biology. We're on Unit 3, Life on Earth, and today we're looking at photosynthesis. That's something you will have heard about and talked about in previous science and biology lessons, but obviously as we go through it, we're going to add some more detail, more complexity at National 5 level. Before we go into our Unit 3 stuff, I just want to remind you back to Unit 2. So Kinnear 2.5, we were looking at the structure of a leaf, because obviously the leaf is an organ and it's the main site of photosynthesis. It contains a huge amount of specialised cells that facilitate that particular job of taking sunlight and making food. And alongside that diagram from Unit 2, you'll have a description of the different cells and structures involved within the leaf structure. So why is a cuticle? It's waterproof, it stops water being lost by evaporation. Upper epidermis, it allows light to go through. This is not where photosynthesis happens. It doesn't have any chloroplasts, but it does allow the light to go through into the palisade mesophils. This is where the magic happens. We're going to talk about what happens in these cells and in the chloroplast today. Below that is the spongy mesophyll, and they called spongy mesophyll because they have lots of gaps and air spaces in between them, and that allows the gases to circulate. And obviously there's two important gases involved in photosynthesis, which we'll talk about. Also involved are the guard cells and stomata. The guard cells are the sausage-like structures that open and close the pore, the stomata. That again controls the entry and exit of carbon dioxide, oxygen, and water, which again are obviously very important within the process we're talking about today. And lastly, the vascular bundle. So again, we've talked about xylem and phloem. So xylem being important in getting water from roots to the leaf for photosynthesis to happen. Again, once it's done, then you've got glucose being produced. So you want to transport that to all parts of the plant, and that's done via the phloem. So all of these individual parts that we talked about in Key Area 2.5 in Unit 2, they're obviously really important to what we're going to talk about today. So make sure you check back on your notes and you relate these things to what you're learning about today. So today we're talking about the chemistry of photosynthesis. You will have talked about this and seen this in your science and biology lessons previously. You'll know that the sun is involved. We've talked about that being involved, obviously, when it comes to food chains and food webs and the flow of energy. But today we're talking about how that gets into the system, and that's through photosynthesis. It's a series of enzyme control reactions. We previously would have given you a chemical formula, which is one thing happening, but actually it's a series of things, and we see it happening in two stages. The first one requiring light, so that's a light-dependent stage, and that's photolysis. So you should be able to identify from that word photolysis the two parts being photo, which means light, doesn't mean taking a picture, photo meaning light, and lysis meaning break. So we're going to talk about something being broken by the use of light. And stage two, carbon fixation. It's not light dependent. It doesn't require light. It can happen in the dark, but it is a temperature dependent stage, and that relates back to this enzyme controlled reactions. And as we know, temperature has an effect on enzymes. So again, we'll talk about that as we go through the process. So in photolysis, our stage one, the light energy is trapped by chlorophyll. So obviously that is a chemical pigment which you find within chloroplasts, and it's converted into chemical energy. So that makes it useful and usable by the plants themselves, but obviously the animals that come along and eat those plants. Light energy is used to split water molecules into its constituent parts, hydrogen and oxygen. And oxygen in this process is a waste product, so obviously plants release it, which is great for us. We're getting food and oxygen, two things that are vital for our life from this plant process. Let's put photolysis into diagram form, and I'll talk you through it as we go. So light energy from the sun is then trapped by chlorophyll, this green pigment that we find in chloroplasts. It's the stuff that makes plants look green. That energy is then used to break water, H2O, into its constituent parts, oxygen and hydrogen. Oxygen is a waste product, so that is released. It will diffuse out of the palisade mesophyll into the gaps between the spongy mesophyll and eventually out through the open stomata. Hydrogen, however, is going to be useful. We're going to need that for stage 2, but it's also a very reactive element, so can't just be left to zoom about the cells on its own. So we're going to get a hydrogen carrier, which is going to reduce, grab hold of that hydrogen and take it onto stage 2. The energy we use to break water is also used to make ATP from ADP and PI. So again, that ATP is an energy currency. So we've now got that to move on to stage 2 as well. Just in text form, the hydrogen joins with a hydrogen carrier. At this stage in National 5, you don't need to know the name of that molecule. You will learn the name when you go into higher biology next year. Hydrogen is then taken by that carrier to the next stage, carbon fixation. We also make ATP during photolysis. That's then called photophosphorylation. So the process of adding phosphate onto ADP to make ATP is phosphorylation, and because it's done by light, that makes it photophosphorylation. And that ATP is also used in stage 2, which we'll go on to next. Carbon fixation, as I said, is a second stage. It also occurs in the chloroplast. It doesn't require light, however, but it is controlled by enzymes and therefore is temperature dependent. We're going to use the hydrogen and the ATP from the first stage. We're also going to bring in carbon dioxide from the air, which obviously has been diffused in through the stomata and in through the spongy mesophyll into our palisade mesophyll cells, which is where our chloroplasts mainly are. We're also then going to combine that carbon dioxide and the hydrogen to form glucose. Carbon fixation in diagram form then. We've got hydrogen brought by the carrier molecule from photolysis and carbon dioxide from the air. So that again has diffused in through the open stomata into the air spaces between spongy mesophyll and eventually into the palisade mesophyll, where this is happening in the chloroplasts. And those two are put together to make glucose. But making chemical bonds requires energy, and that energy comes from our ATP, our energy currency. That again has come from photolysis. So that's broken down into ATP plus PI. That provides the energy to build the glucose molecules from our hydrogen and carbon dioxide. Now that the plant has successfully made glucose from photosynthesis, it can use it in 3 different ways. Firstly, immediate energy source for things like growth and reproduction, releasing the energy by respiration. It can also store it as starch, so starch being a complex carbohydrate made from glucose. And when it needs glucose, it can convert it back as and when it's required. And that's why we like eating plants, because it obviously has that complex carbohydrate and starch. We can obviously convert it ourselves back into glucose or other sugars. It can also be used as a structural carbohydrate. So again, cellulose, which we've talked about being parts of the cell walls in plants. So that's another use for it. We've talked about the chemistry of photosynthesis. It's a series of chemical reactions, and therefore those chemical reactions can be altered in terms of the rate at which they happen. That could be by a number of factors, and they are called limiting factors. The first one is light intensity. So stage 1 photolysis requires light energy to break water. Less light means less ability for that to happen. Stage 2 is an enzyme-controlled reaction or series of enzyme-controlled reactions. So therefore, temperature can impact on the enzymes and therefore the rate of photosynthesis. The last one, carbon dioxide concentration. Again, in stage 2, we're building glucose from carbon dioxide and hydrogen. So again, less carbon dioxide means less photosynthesis can happen. So in the lab, we can measure the rate of photosynthesis by looking at a number of things. So we could look at the amount of oxygen that is released by the plant. We can look at the amount of carbon dioxide that's taken up by the plant. We can also even look at the increased mass. So we know that glucose is used for plant growth and making cell walls. So again, more photosynthesis means an increase in mass of the plant. Here's an example of a graph showing how light intensity affects the rate of photosynthesis. So as you can see, along the bottom we have light intensity. So the light intensity is increasing as you go right. Rate of photosynthesis on the left, if it goes up as you go up the y-axis. So photosynthesis can't happen without light. So even if we've got plenty of water, plenty of carbon dioxide, it's not going to happen without light. So as you can see, increasing the light intensity boosts the rate of photosynthesis. There is, however, a plateau. And as you can see, as you increase the light intensity, the rate of photosynthesis does not increase. That's unfortunately because at that stage, we're then seeing carbon dioxide limit the rate of photosynthesis. So we could increase it by adding more carbon dioxide into the experiment. And a similar graph here for carbon dioxide concentration. So carbon dioxide is a substrate or an ingredient of photosynthesis. So it can't happen without carbon dioxide. So even if there's plenty of light, the planet can't photosynthesize without carbon dioxide. So as carbon dioxide concentration increases, the rate of photosynthesis increases. But likewise, we've got a plateau. And again, that is then where, despite having more and more carbon dioxide, the amount of light available is limiting the rate of photosynthesis. So if we were to increase the amount of light, we could still see an increase in the rate of photosynthesis as we add in more carbon dioxide. So the two of those things work together. In our final graph for temperature, it looks a different trend, but something we've seen before when we've talked about enzymes. So as you can see along the bottom there, temperature is increasing as you go right. And at the first stage, as we increase temperature, we increase the rate of photosynthesis until we get to that peak. And that's a second bit where we've got the optimum temperature. So photosynthesis can't work any faster. Our enzymes are churning out the reactions as fast as they possibly can. And as we've seen with enzymes in previous units, at stage 3, at this rate, photosynthesis is slowing down. As you increase the temperature, you're unfortunately denaturing the enzymes, therefore you're stopping them doing their job, therefore you'll eventually get to a stage where photosynthesis can't happen because we've completely destroyed the enzyme, denatured the enzymes that are doing the jobs within the photosynthesis reactions. If we are able to reduce those limiting factors of photosynthesis, we are then able to increase our yield and produce earlier crops. So that's fantastic if we're putting photosynthesis in the role of food provider for us, putting food on our tables. Managing those limiting factors will have an impact on how much food we get and when we can get it. We can't easily do that in a farm in a field, but if we have controlled environments, so polytunnels and greenhouses, you'll see more and more farms using polytunnels. Maybe in your local environment, you'll have them. They allow for us to manage the limiting factors a lot better. So artificial lighting could also be used to extend the number of daylight hours and the amount of light intensity that we have falling on the plants. We can heat the greenhouses, maintain those optimum temperatures through a variety of seasons, and obviously at night times as well when temperatures normally decrease. And this one kind of goes a bit hand in hand with that. We can also release carbon dioxide from heaters that burn fossil fuels. So there's an example of a paraffin heater that will heat up the greenhouse but also release carbon dioxide. Or you can obviously just pump carbon dioxide into your polytunnel or greenhouse. So by doing that in horticulture, we can increase the yield and produce crops earlier than we maybe could do out in a field in Great Britain, where temperatures and sunlight will be something out with our control. And that's us today. So we have learned that photosynthesis happens in two stages: photolysis and carbon fixation. That happens within the chloroplasts, which are found most abundantly in the palisade mesophyll cells within a leaf. And that's something we learned in Key Area 2.5 back in Unit 2. The rate of photosynthesis can be limited by things like light intensity, temperature, and carbon dioxide concentration. But in horticulture, so putting food on our tables, we can manage those limiting factors and therefore increase the plant yield and produce early crops. So that then helps us feed the ever expanding population. So that's how photosynthesis then relates to food production. So hopefully you found that useful and informative. As always, if you have any problems, questions, or queries, let me know, and I'll speak to you again soon. Bye.